What if the solutions to humanity's biggest challenges were hiding in the smallest of places? Now let's think small, really small. Imagine a world teeming with life that's invisible to the naked eye. This is the world of microbiomes, vast communities of microorganisms that reside in, on, and around us. Microorganisms, including bacteria, fungi, and viruses, work in harmony to create these intricate ecosystems. Each one of us is a walking, breathing universe of these tiny life forms. They play a vital role in our health, from assisting in digestion to defending us against harmful invaders. But they also contribute to diseases like Alzheimer's and asthma when their balance goes awry. Even beyond our bodies, microbiomes in farm animals and soil contribute to global phenomena, such as climate change. As it turns out, these microscopic communities might just be the key to solving some of our most daunting issues. Enter CRISPR, the revolutionary technology that's changing the game. Imagine the most intricate word processor, capable of finding, cutting and pasting not just words or sentences, but the very letters of life itself, DNA. This is what CRISPR technology does. It's like a genetic word processor. CRISPR, which stands for Clustered Regularly Interspaced Short Palindromic Repeats, is a naturally occurring defense mechanism found in a wide range of bacteria. As a tool, it has been adapted by scientists for use in a broad range of organisms to precisely target and cut any sequence of DNA. This technology is inspired by the way bacteria fight off viruses. When a bacterium encounters a virus, it can store a bit of the viral DNA in its own genetic code in a DNA archive called the CRISPR sequence. When the bacterium encounters the virus again, it uses this archive to create RNA segments that match the viral DNA. These RNA segments, along with a protein called Cas9, form a complex that can identify and bind to the viral DNA. The Cas9 protein then acts like a pair of molecular scissors, cutting the viral DNA at just the right spot to disable the virus. This is the cut part of the find, cut, and paste mechanism of CRISPR. Scientists have harnessed this mechanism for gene editing. By designing RNA sequences that match a specific location in an organism's DNA, the CRISPR-Cas9 complex can be directed to cut the DNA at that exact spot. Once the DNA is cut, the cell's natural repair machinery kicks in to fix the break. Scientists can use this repair process to paste in a new gene or make other changes to the DNA sequence. CRISPR has already made waves in the scientific community with its noteworthy applications. It has allowed us to cure debilitating disorders like sickle cell disease and create drought and disease-resistant rice plants, showcasing its potential to revolutionize medicine and agriculture. But the real game changer? CRISPR's potential to edit entire microbiomes. This opens up a new world of possibilities, from fighting diseases tied to our gut bacteria to tackling climate change by modifying the microbiomes of livestock. Indeed, the future of CRISPR technology seems boundless. You see, microbiomes aren't just bystanders. They play an active role in our health. The complex interactions within these microbial communities are critical to our well-being. However, when these microbiomes malfunction, they can contribute to a host of diseases. For instance, Alzheimer's disease, a debilitating neurodegenerative disorder, has been linked to dysfunctional gut microbiomes. Similarly, asthma, a chronic respiratory condition too, has ties to irregularities in our microbiomes. But the influence of microbiomes doesn't stop at human health. They also play a significant role in our environment. Consider farm animals. Their gut microbiomes are responsible for producing methane, a potent greenhouse gas that drives climate change. Yes, these tiny microorganisms within farm animals contribute to the warming of our planet. Understanding and addressing these intricate microbiome interactions can open doors to potential solutions for various health and environmental issues. Clearly, our tiny co-inhabitants have a big impact. This is where precision microbiome editing steps in. Imagine a new field of science that harnesses the power of both CRISPR and metagenomics, a tool for studying microbial communities. This is precision microbiome editing, a cutting-edge approach that allows us to not just understand, but also manipulate the complex world of microbiomes to our benefit. Let's break it down. CRISPR allows us to precisely edit DNA, like a sophisticated word processor for genes. Metagenomics, on the other hand, gives us a detailed picture of entire microbial communities, without needing to isolate and culture individual species. When we combine these technologies, we get a powerful tool that lets us identify links between dysfunctional microbiomes and diseases or greenhouse gas emissions, develop tailored microbiome editors, and deploy safe and effective solutions. Now let's talk about potential. Consider methane emissions from livestock a significant contributor to climate change. Precision microbiome editing could help us reduce these emissions by up to 80%. How? By modifying a calf's microbiome right at birth. This could have a dual benefit. It would help protect our environment, and it would also benefit farmers by improving the health and productivity of their livestock. But the potential of precision microbiome editing extends beyond the farm. Imagine being able to offer non-invasive therapies for children at risk of asthma, or to treat, or perhaps even prevent other microbiome-linked diseases like obesity, diabetes, and Alzheimer's. The possibilities are staggering. So, we see that precision microbiome editing is not just about understanding the complex interactions within microbiomes. It's about using that understanding to create targeted interventions that can help tackle some of our biggest challenges, from climate change to public health. The implications are far-reaching, from combating climate change to improving human health. Through precision microbiome editing, we can harness the power of microbiomes, and in doing so, unlock transformative solutions to some of the world's most pressing challenges. This is the promise of precision microbiome editing, the promise of a brighter, healthier future for all. Imagine a world where we work hand in hand with nature to solve our biggest problems. A world where we use the precision of CRISPR technology not only to cure devastating diseases but also to edit the very essence of life, the microbiomes. These miniature ecosystems teeming with diverse microorganisms are integral to our existence, influencing everything from our health to our environment. By harnessing the power of these microbiomes, we can mitigate climate change, reduce methane emissions from livestock, and even prevent diseases like Alzheimer's and asthma. This is the transformative potential of precision microbiome editing. It's a new field of science that combines the revolutionary CRISPR technology with metagenomics, allowing us to understand, optimize, and ultimately edit these microscopic communities. In this unexpected collaboration between humans and microorganisms, we find our key to a brighter, more sustainable future.